In this video lecture, we will be discussing on topic that is enumeration of microorganism in the air. First of all, we will start with the introduction. The number and the varieties of method which has been proposed for the enumeration of microorganism. There are the various method to enumerate the number of microorganisms which are present in the air. On the basis of their mode of action, aerosol sampler may be classified as follow. Aerosol means the biological contaminants suspended in the air are referred to as an aerosol. Aerosols means the biological contaminant which are suspended in the air, that is referred to as aerosol. The size of the aerosols that vary from 1 micron to 50 micron or possibility more. So there are the various methods for the enumeration of microorganisms which are present in the air. The method are as follows: impingement in the liquid, impaction on solid surface, filtration, sedimentation, centrifugation, electrostatic precipitation, and the last, that is thermal precipitation. We will study all this method in a detail one by one. First, that is impingement in the liquid. First method, that is the impingement in liquid. Impingement means something that impinged and onto the liquid surfaces. On what? In the liquid surfaces. Okay. The first method, that is name is impingement in the liquid. Whatever the air containing microorganisms, okay, whatever the air that will impinged onto the liquid surfaces. The entire figure is of lemon sampler. This figure is of lemon sampler. The device name is lemon sampler. This lemon sampler consists of glass fouling aeration tube. This is the fouling aeration tube. Here it has been written, this is the glass fouling aeration tube. Okay. It has been, there is the end, there is the bulb. You can see here it is a bulb. Okay. At the end of the fouling aeration tube, there is the bulb by the 6 holes. There is the 6 holes are there at the end of this bulb. This is the fouling aeration tube. At the end, there is the 6 end perforated holes are there. This tube is passed through the 2-hole rubber stopper. This fouling aeration tube is passed with the 2-hole rubber stopper. Suppose this is the rubber stopper and it has been passed through this 2-hole rubber stopper. One, that is the fouling aeration tube, and the second one, that is the Zelda trap. Second one, that is the Zelda trap, and from one hole there is the fouling aeration tube. It is being passed. In the lemon sampler, there is the fouling aeration tube is passed through this one of the rubber stopper, and the second, that is the Zelda trap. In the Zelda trap, there is this square glass baffles are there. Square glass baffles, what is the function of the square glass baffles over here? That is being inserted with the help that will break the clump of aerosol. Whatever the clump that is being present in the biological contaminant suspending air, if it is a clump it is present, that will break those clump. So this is the function of the Zelda trap. And there is this some small flow meter is there that will measure the rate of the air flow entering the upper open end of the fouling tube. Whatever the air that is coming, that will measure and that will pass through the fouling aeration tube. The entire sampler, this entire sampler can be sterilized by the autoclaving, or we can also sterilize by the rinsing with the 70% of alcohol, and then we have to dry. So this entire device, or you can say the sampler, can be sterilized by the autoclaving or by 70% of rinsing with 70% of alcohol, and then we have to go for drying. Whatever the air coming from the flow meter that will pass through the fouling aeration tube, Okay, now at the end of the Foley narration bulb, there is the 20 ml of the ant broth, it is there. 20 ml of the ant broth containing this tube, and 2 to 3 drops of olive oil is there. The sterile olive oil is there to prevent from the foaming. The function of this olive oil, 2 to 3 drops of olive oil, that will prevent from foaming, and 20 ml, this much mark, that is filled with the ant broth. Now this broth containing whatever the microorganisms come from the air, that will settle in this broth, and now we have to take this aliquot from this broth, okay, and we have to incubate into the plate. Okay, we have to incubate this aliquots onto the agar plate, and we have to incubate this plate and observe for the colony, and we have to count the colonies which are present. Okay, that is all about the impingement in the liquid. It is a very simple, just we have to remember that the entire figure, this is the figure name is lemon sampler, or you can say the device, that is a lemon sampler. Okay, from here the air will pass, the flow meter will measure the amount of air that is coming. Okay, the air will pass from the Foley narration tube. Okay, and at the end of the Foley narration tube there is the 6 hole. Okay, whatever the air that is coming, that will come onto these, and it is here, up to here, it is filled with the 20 ml of ant broth. Okay, ant broth it is there, and 2 to 3 drops of olive oil. Olive oil will prevent from foaming. Okay, then from this, whatever the air it has been, now it has been entered into this ant broth. Now we will take from this aliquot and we will transfer into the agar plate. After transfer into the agar plate, we will incubate this agar plate for the particular time period and the hours, and after that we will count the colonies. Okay, so this is what all about the impingement in the liquid. Another method, that is the capillary impingement. Okay, there is also the another method for the impingement in the liquid, which is named as capillary impinger. Okay, capillary impinger, it is also the another method for the impingement in the liquid. Okay, the entire capillary impinger is given by the scientist Kluwer and Wissler. Okay, this has been given by the Kluwer and Wissler, and it is the most efficient, simple and satisfactory method. It is one of the most simple, efficient and satisfactory method. In this method, air is brought in a contact with the water through the capillary tube and break up the clump of the bacteria. From here the air will come, okay, through this capillary tube, and whatever the bacteria, clump of bacteria, it will break those clump of bacteria, and now this air enter into the water. Okay, this method brought in a contact with the water. Okay, here it is, the water is there, the air come from this direction, and whatever the air containing microorganism, that will impinged into this liquid. Okay, here it has been, liquid is there, liquid containing water. Now we have to take this aliquot. Aliquot of a water is mixed with the melted and nutrient agar in a petri dish. Okay, now we have to take the aliquot from this water, and now from that we will place onto the agar plate, and then we will place this agar plate for the particular time and the hours, and after that incubation the colonies are counted. From the colonies we can determine that this number of microorganisms are present in the particular air environment. So first method, that is the impingement in the liquid. In the impingement in the liquid, we have studied the two method. First, that is the lemon sampler devices, and the second, that is the capillary impingement. Okay, in the lemon sampler, that is the 20 ml of broth that is filled. Okay, from that broth we take the aliquots and further we count the colonies. Whereas in capillary impingement, what we are doing, this is the instrument. Okay, this is the device, air is coming, okay, from the capillary tube, it will impinged onto the distilled water, or thus water is present. Okay, distilled water. Okay, from this water now we will take the aliquots and the count are made. Now the second method, that is impaction on solid surfaces. Okay, impaction on solid surfaces. First method name was impingement in liquid. Whatever the air coming that will impinge onto the liquid surfaces. Now the second method, that is impaction on solid surfaces. Whatever the air that is coming that will impact onto the solid surfaces, not onto the liquid surfaces. Sampler of this type in which deposited onto the suspended particle directly onto the solid surfaces were observed. Directly whatever the air coming that will impinge onto the solid surfaces. The organism are generally deposited onto the agar medium. Okay, here there is the agar, whereas in the liquid there were the and broth were there and the distilled water were there. Okay, but here there will be the agar will be there at the for the deposition, and the agar is incubated. Now we will incubate this agar until the visible colonies appear and the count are made. Okay, whatever the air coming that will impact onto the solid surfaces, that will deposit it onto the solid surfaces such as agar. Okay, agar containing those things. Now this agar containing plates were incubated for the particular time hours. Okay, and then after we will count the colonies. Okay, this is the figure of Hola and the Dalla Valle sampler. Okay, this entire figure it is of Hola and the Dalla Valle sampler. Okay, this container, the entire container that you are seeing, okay, this entire container or a sampler you can say, that is consist of a brass. It is made up of brass material. Okay, it is made up of brass materials with a removable bottom. Here there is the bottom, is here the bottom it is there, it is a removable, we can remove this bottom. So this entire container or a sampler, it is made up of brass, it is made up of brass and it is consist of removable bottom. The bottom we can remove. Okay, the container, this container or sampler is fitted with an inverted glass funnel. You can see this is the funnel, but it is inverted. Okay, it is a fitted with an inverted glass funnel, and this glass funnel that sit approximately half from the bottom of the Petri plate. This is the Petri plate, you can see this is the Petri plate. Okay, and in the half of the Petri plate that sit, okay, that is been fitted as if like such a way that that does not touches the Petri plate, but half of the Petri plate it touches. Okay, it is such a way. Now this Petri plate, here this is the Petri plate, this containing the nutrient agar, this Petri plate containing the nutrient agar that is being placed in the lower portion of the container, of the entire container it is being placed at the lower portion, and then it is been screwed. This Petri plate it is screwed tightly against this gasket. Okay, this gasket will control, or you can see that that will fix up this Petri plate. Okay, and this Petri plate is being placed into the lower portion of this container. The inside of the funnel or a rim, or you can say this is the funnel rim, this is being swabbed, that is been cleaned with an alcohol before the use. When before the use we will just clean this rim portion, okay, with the help of alcohol. Now what happened, the air sampler, whatever the air coming from here, okay, from the funnel steam, whatever the air that will impinged onto the agar medium containing an agar. Okay, and then we have to remove this plate after the particular time hours. We have to remove this plate and we have to place this an agar plate into the incubator for the particular times hours and the minutes. Okay, for the particular time hours and the particular temperature we have to put this, and after that we count the number of colonies. Okay, so we can say that this method is a simple portable and efficient method for testing the carried out in a one operation. Okay, this device name is Holla-ended Delavalle sampler, which is made up of brass. This entire container or sampler, it is made up of brass, and that is the inverted funnel. It is being seated in a such a way that half of the petri plate, and the petri plate, it is placed into the lower portion of the container. Air coming from this funnel, and then it is impinged onto the agar, nutrient agar, and now we have to take out the nutrient agar, as there is a removable bottom. Okay, we can remove the bottom, and we take out the petri plate, and after taking out the petri plate, we can place for the particular time hours, and after that the visible colonies will develop, and we will count the numbers of colonies. Okay, so this is the method which is known as impaction on solid surfaces. Okay,